Hello, biologists. Today we're going to talk about mitosis and the cell cycle. First, let's discuss the goals of mitosis. The goal of mitosis is to create two identical cells. So you're taking one cell and you're duplicating it into two. It creates diploid somatic cells. Let's talk about that vocab. So diploid means that there are two sets of chromosomes. Humans are diploid. We get one set of chromosomes from our mom and one set of chromosomes from our dad. So two sets of chromosomes. That doesn't mean that there's two chromosomes. It means there's two sets of chromosomes. In humans, we have 46 chromosomes total. Chromosomes total. 23 from mom. And 23 from dad. So that's how you get 46. Somatic cells refer to the fact that they are body cells. That's going to be your hair, your skin, your muscle, your brain, your liver, everything in your body except your sperm and your egg. Those are not somatic cells. We refer to those as sex cells. So everything else is somatic. The word somatic literally means body. So anything from your kidneys to your toenails, that's somatic. Mitosis is also going to be used to grow and repair and develop. So if you think about the fact that you look so much more different now than you did when you were 3, all of the tissue that you created, the height and weight you've put on, all of that is because of mitosis only. From the moment you were one single cell, you divide into 2, and then into 4, and then into 8, and then 16, and then you were a fetus, and then you were a baby, and then you were a small child. All of that is mitosis. Mitosis is used for everything except producing sperm and egg. We also use mitosis for growing our hair, growing our skin, growing our fingernails. We are going to create red blood cells. You are creating 3 million red blood cells every second because they have a short little lifespan. So we're constantly making them. You're going to use mitosis for any damage repair. So if you have a paper cut or a bruise, mitosis will repair that. It's literally everything. And if you're another type of organism other than a mammal, you can use mitosis for asexual reproduction. So plants do asexual reproduction a lot. So do fungi. So do bugs. Some lizards do. It's not uncommon in the animal kingdom, in the plant kingdom, and the fungi kingdom. But it is not something that mammals do. Mammals do not do it, as far as we know. The way that asexual reproduction works is you basically make a genetic clone of yourself. Lichen do it with a process called fragmentation. So they make these little finger-like projections, and then they break one off, and that grows into a whole new individual. They just fragment off into a new lichen. So that would be mitosis as well. There is no change in the genetics. That's what we mean by identical cells. How does mitosis work? Let's talk about it. The first thing that we need to do is take our 6 feet of DNA, our single-stranded double helix right here, and we need to make it compact so that we can move it around our cells. To make it compact, we're going to take our DNA and we're going to wrap it around some special proteins called histones. Histone proteins we'll talk about later in the term too. They're important for a process called epigenetics, and they are really important to packaging our DNA so that we can move it without it getting tangled and broken. So your 6 feet of DNA in every single nucleus is going to be tightly woven around these histoproteins. And when you have DNA wrapped around histones, you call it chromatin. So chromatin is DNA wrapped around a histone protein. Here's your DNA. This is going from smaller to larger. This is a histone protein in the center. And when you have DNA wrapped around histones, we call it chromatin. Chromatin will coil up into these little tight packages, and then those coils coil into supercoils, and those supercoils package together into a visible chromosome. So here we have chromatin spiraling up, packaging itself into tight little flat flower shapes. Those compress and compress, and that is how you condense it enough to get a chromosome. DNA is not condensed into chromosomes until your cell is going to divide. If your cell is doing regular self-processes, which is where it spends 90% of its life, the DNA is loose, it's not packaged up. We only want it tightly coiled up so that we can move it between the cells that we're creating without losing any of it. So we say that you have 46 chromosomes. We know that because we can count them when they become visible during mitosis. But those chromosomes don't exist inside your cell until it's about to do mitosis or another cellular processes where it divides. So those chromosomes, the DNA is always there, but they're not packaged until you need them to be packaged. Here is a single chromosome. It's condensed. It's made of DNA wrapped around those histone proteins. So this chromosome is composed of chromatin. In the center of your chromosome, and it's not the exact center, there's a short arm and a long arm, there is a pinched in portion called the centromere. The centromere is a series of connection proteins. Those connection proteins will attach one copy of a chromosome to an identical copy of a chromosome. And those proteins collectively are called the kinetochore. Kinetochore. I know it looks like it says chore, but we don't pronounce the H. So kinetochore. Those kinetochore proteins have little proteins attaching these two halves together. And those are called adhesion proteins because they're adhesive, right? So those adhesive proteins hold them together. And the kinetochore is also important because it allows your chromosomes to attach to the spindle fibers, which will move them around the cell. Spindle fibers are microtubules, and they are composed of the cytoskeleton. These little attachment points here allow your cell to like tug of war back and forth on that chromosome in the center and move it. It's how we control where it's going, and we need to pull it into opposite halves of the cell so we can divide the cell into two. When you have a single chromosome, you want to duplicate your cell into two, so you need to copy that chromosome. When you have two copied chromosomes, you hold them together so you can make sure that you don't lose one. They're held together at the kinetochore, and we refer to each of these as a sister chromatid. Now, note that they are identical. They are exactly the same. They are copies of each other. We do not call this two chromosomes. And the reason we don't call it two chromosomes is because it's one chromosome copied. So it's double the amount of DNA, but we identify each chromosome by the kinetochore. So this is one chromosome. You just have double the amount of it. Here's another visual. This is a chromosome. It has the short arm and the long arm. Here's the centromere, which is the pinched in part in the center. When it duplicates, you have both of your chromosome halves held together. This chromosome one of them here, one of them here, just a chromosome, but it's duplicated. And it's held together at the centromere with those proteins called the kinetochore. Here's another visualization. We have the short arm and the long arm. Here's the pinched in part in the center. That's your centromere. We have duplicated this one chromosome, so we have double the chromosome information, but it's still a chromosome. And in the centromere, you have those kinetochore proteins and adhesion proteins holding the two sister chromatids together and allowing the sister chromatids to attach to the microtubules of the spindle. We also have the telomeres identified on a couple of these images. The telomeres are like little caps on the end of your chromosomes to protect them from unraveling. So they're little protective tops. When your cell is going to do mitosis, it's going to follow 5 basic steps. So we're looking at this right here. The first step is going to be interphase. In interphase, your cell is doing its normal life processes. It's not trying to divide. The chromosome is decondensed. I can often see a nucleolus in this phase. So this is a cell living its life. In interphase, the cell will go through 3 different steps. So here we have the cell cycle up in the top left. Interphase, which is most of the cell's life, is composed of G1, S, and G2 phase. So interphase is not part of mitosis. It is the regular life process of the cell. In interphase, your cell will be preparing for mitosis. So in G1, the cell will start to grow. It will duplicate its organelles. In S phase, you're going to synthesize the DNA. So this is where the DNA gets copied. Think S for synthesis. And in G2 phase, your cell's going to get even bigger. It's going to make sure everything is duplicated. And then if it's ready, it will go through mitosis. It will do mitotic phase. In mitosis, there's 2 parts. There's the actual creating 2 copies of your nuclei, making sure you have duplicate information of everything and that it's identical. And then the last part is cytokinesis, which is also technically not part of mitosis. So cytokinesis, if we break it down, cyto is cell and kinesis is movement. That's when we physically pinch the 2 cells apart and pull them apart. So it's the movement of the cell, the movement of the plasma to create 2 cells where there's 1. I like to think of mitosis as duplicating the nucleus, and cytokinesis is dividing the cytoplasm. In the mitosis process, you have your regular life, chilling as a cell. Everything's decondensed. And the first step of mitosis, when your cell decides it's actively going to split into 2 identical cells, we have our first movement of the chromosomes, and that's prophase. Pro means first. So in prophase, your DNA condenses, it becomes visible. It was already duplicated during interphase, so I have 2 copies of each chromosome. The 2 copies are referred to as sister chromatids, and they are held together at the centromere. That's that white dot. So the centromere has your kinetochore and your adhesion proteins. We also are going to duplicate our aster, or our spindle apparatus, or our centrioles. Those are synonyms for the same thing. I like the word aster because that's a type of flower. A sunflower is an aster, so I think it has like beautiful petals coming out the side, and that's kind of what it looks like to me. Spindle apparatus also makes sense too because it becomes spindly with the microtubules coming out of it, so both of those are nice visual words. In prophase, we also lose our nuclear envelope. Note that the nuclear envelope has disintegrated. So now we have condensed DNA. It's packaged into chromosomes that are held together at the kinetochore, and those are going to be free to move around the cell now so we can make sure we get 1 of each of those DNA copies into our new cells. The next step of mitosis is metaphase. Think meta for middle. In metaphase, your sister chromatids line up on this metaphase plate, which is basically in the center. Think of it like the equator of the cell inside, right in the center. Each one of those moves into the center of the cell because it's being controlled by the spindle apparatus. So here's a spindle fiber. It's attached to the kinetochore of my sister chromatids, and it pushes and pulls like a tug of rope until it moves it into the center of the cell. Now that my cell has done metaphase, I'm going to do anaphase. Think ana for away. In anaphase, the spindle apparatus shortens, so it pulls on those microtubules. At the same time, the cohesion and adhesion proteins that were holding your sister chromatids apart disintegrate. And so my sister chromatids are separated. Now I have 1 chromosome on each side, and they're moving toward the opposite poles. So I've lined up everything I want to duplicate in the center, and I've pulled each copy in half. So I have 1 copy on the top and 1 copy on the bottom. In telophase, if you say telo, it's okay, but most people say telo. In telophase, you're going to have the cytoplasm start to pinch in, but not fully separate. Your nuclear envelope is going to reappear. your chromosomes are going to start to decondense because you want to be able to access your DNA in your new cell. And the last step, which is technically not part of mitosis, is called cytokinesis. In the last step, you're going to have some proteins that come in and they snip the two cells apart, and you will divide the cytoplasm so you have two fully separate cells. Let's look through that process again. Here I have a single cell. Here are these centrioles, the centromeres, centrioles, spindle apparatus, whichever you like to call it. I have my decondensed DNA. I can still see the nucleolus, which is the part of the DNA that we use to make ribosomes. We just put it all together and it stains dark. So we can tell that the cell is still in interphase because none of the chromosomes have condensed. This is when your cell is doing interphase steps 1, 2, and 3, which is G1, S, and G2. During prophase, these two steps are going to be prophase together. Your spindle apparatus starts to push out its little microtubules, starts to look like a star, so it looks like an aster. The nuclear envelope starts to dissolve and disappear, and your chromosomes appear. The DNA is condensing around the histone proteins, which are wrapping up tightly into chromosomes. I've already duplicated them during S phase of interphase, so I have 2 copies of each chromosome held together at the centromere with the kinetochore. Those adhesion proteins in the kinetochore are holding my 2 sister chromatids of this one chromosome together. So here I have 4 chromosomes, each one composed of 2 sister chromatids, each sister chromatid being identical to each other. So we're not creating any new genetics here. We're just duplicating what we already have, creating identical cells. When we continue with prophase, the asters are going to move to opposite poles. The chromosomes will be fully condensed, held together at the centromeres. And then each of these little asters will reach out and it will connect to the kinetochore and allow it to push and pull the chromosomes into the center of the cell. When they're in the center of the cell, we refer to it as metaphase, meta for middle. In metaphase, my sister chromatids are lined up. I have 1 copy on each side. The adhesion proteins are still holding them together, but each of my microtubules from the spindle fibers has attached to the kinetochore so that when those adhesion proteins break apart, I can separate my sister chromatids, and each of my new cells will have 1 copy of the chromosomes that I started with. In anaphase, those adhesion proteins break, the spindle apparatus starts to shorten, it pulls the chromosomes apart, so I no longer have sister chromatids. So this is a chromosome and this is a chromosome. These 2 chromosomes are identical to each other, and they used to be held together, but I've just separated them. I'm pulling them toward opposite ends of the pool using my spindle apparatus fibers, which are made of microtubules. In anaphase, the cell will start to pinch off. The chromosomes will move toward the poles. And in telophase, I will start to reset my cells. The cell will start to contract even more. The nuclear envelope will reform. The spindle apparatus will go away. And my cells will start to decondense and turn back into loose DNA so I can use my cell and do whatever it wants to do. Here, I have a fully formed 2 new cells. I started with 1 cell. We refer to it as the mother cell. I now have 2 daughter cells. Both of them have identical copies of DNA. They are identical to each other. They both have 2 sets of DNA, 1 from each parent. So both of these are diploid. And these cells are somatic. They're body cells. It could be a liver cell, it could be a bone cell, whatever it is. I have cut the cytoplasm, which means that cytokinesis has occurred. One more visual for you. No chromosomes, indicating this is interphase. I have my nuclear envelope, so I know that I'm an interphase. Those are my key things. In interphase, I'm going to duplicate the DNA. In prophase, the DNA will start to condense into chromosomes. The nuclear envelope will disappear because it would be in the way. We need to get rid of it. The spindle fibers start to show up and move towards the poles. Some of your books like to add prometaphase as its own step. That's totally cool. It's just an intermediary between metaphase and prophase. So in prometaphase, the spindle fibers attach to the kinetochores and they start to push and pull the chromosomes toward the metaphase plate. In metaphase, the chromosomes are lined up on the metaphase plate. They are held together at the centromere using the kinetochore adhesion proteins. Each side of this chromosome is a sister chromatid. The two halves are identical to each other. The spindle fibers will shorten and pull the sister chromatids apart in anaphase. And then in telophase, everything starts to reset. So the cell starts to divide its cytoplasm, the nuclear envelope reforms, the spindle apparatus goes away, the chromosomes decondense. And when you are done, you can do cytokinesis, and you are left with two identical diploid somatic cells. Here's a visual of cytokinesis occurring. Now in animal cells, this is one half, this is another half. There is something called a contractile protein, which picture a lasso, and then the lasso is being tightened. That is pinching this off. It is literally slicing it in two by contracting and contracting and contracting, and it creates what scientists lovingly call the cleavage furrow. So animal cells have this distinctive cleavage furrow being like the furrow in your eyebrows, that's being a ridge, and then cleavage because it's cleaving it in two. Or you can think that it kind of looks like a butt, and so it has cleavage. Either way, but it's a cleavage furrow. In plant cells, they do not use the same type of contractile proteins because plant cells have a cell wall. The cell wall is rigid. You can't slice it with a wire like you can the animal cells. And so instead of a cleavage furrow, plant cells create what they call a cell plate. So right here, I can see a new cell wall forming, and that cell wall will eventually connect to both sides and thicken and become dense, and that will divide this cell from this cell, and I'll have two cells where I had one. So in animals, we have a cleavage furrow, and in plants, we have a cell plate. The next thing to discuss, now that we've done the steps of mitosis, is the cell cycle in detail. So the cell cycle is composed of interphase and mitosis and cytokinesis. It's all of those three things together. Interphase itself has 3 steps, but there's also checkpoints that we need to discuss. And there is a lot of different molecules and enzymes involved in regulating the cell cycle. The reason the cell cycle is important is because it helps make sure that when we divide our cells, that we're not doing it wrong. Because if we do it wrong, it can lead to problems. Uncontrolled cell growth is what we call the disease cancer. And so we want to make sure that when our cells duplicate, when they're growing, we know exactly what they're doing and that they're not mutated. Let's start right here. In G1, which is the first of the 3 phases of interphase, your cell is going to duplicate its cellular components, everything except DNA. It does that in the next step. It's going to duplicate the endoplasmic reticulum and the Golgi and the lysosome. The mitochondria duplicate on their own. The chloroplasts duplicate on their own. But everything else that's in your cell will be duplicated. It's also going to grow. I think G is for growth. If your cell is doing this and it decides that it's not ready to continue, it can go into a resting phase called G0, where it kind of just sits there and hibernates. So that is an option. But if everything's going well, then your cell will continue with S phase. In S phase, you're going to duplicate the DNA. So each of the 46 chromosomes is duplicated. Think S for synthesis. The last step of interphase is G2. In G2, the cell double checks everything, specifically making sure that it duplicated the chromosomes appropriately. And it's also going to grow bigger. It has to be large enough because if you don't have enough cytoplasm to divide into 2, then there's no point in doing mitosis. So you're going to get even bigger in G2. And if everything looks good, you'll go through your prophase, prometaphase, metaphase, anaphase, telophase, and then lastly, cytokinesis to give you 2 identical somatic cells. The checkpoints in the cell cycle are really important to making sure that we're not making mistakes and growing non-functional or maybe even dangerous cells. Here we have the beginning of G1 and then S and G2. So we have 2 checkpoints in interphase. The first checkpoint in interphase is called the G1 checkpoint. In the G1 checkpoint, your cell's going to double check. Do I have enough nutrients for this? Do I have the appropriate growth factors? Is my DNA okay? Because if it's not okay, I don't want to duplicate it. If any of these are a no, your cell will move over to G0. It will hopefully get repaired, and then eventually will move back into G1. Then your interphase step of S, you will synthesize the DNA, you'll duplicate the DNA, you'll go into G2, and you will make sure that you're big enough and that all your organelles have been duplicated. And lastly, before you enter the mitotic phase, you'll do the G2 checkpoint. You're going to make sure that you're large enough, and you're going to make sure that when you did replicate your DNA, it replicated appropriately. So here we want to make sure it's good before we replicate it, and here we make sure it's good after we replicate it. Then your cell will do mitosis, and at the metaphase checkpoint, it will stop. And it will look, and it will make sure that each one of those sister chromatids is attached to the spindle. Because if I have 2 sister chromatids attached to the spindle, and one of them breaks off, and then in anaphase, this is not attached, I won't be able to separate my sister chromatids. Instead, I would take both sister chromatids toward one cell and none toward the second cell. So I can end up with an inappropriate number of chromosomes. We're going to talk about that when we talk about mutation, but it's called aneuploidy, where you end up with too many or too few chromosomes, and it's actually pretty common. So those are our 3 main cell cycle checkpoints and what each one is looking for. Now, it turns out at these checkpoints, there's different enzymes and molecules involved, and they are so cool. The first one is called cyclins. So cyclin, that's the molecule we're discussing. Cyclins are going to promote going through. So they are, yes, let's divide. The way that they work is they are a group of enzymes, and they turn on other enzymes, and those other enzymes promote cell growth. Cyclins are going to turn on an enzyme called cyclin-dependent kinases. That's the next slide. So cyclins turn on kinases. These cyclins appear in different concentrations throughout the process of cellular division. We have a different type of cyclin for each step of the process. And you can see that in G1 phase to S phase, the G1S cyclin becomes really high in concentration right between those 2 steps. So you can think of it as like a promoter. It's standing there going, yes, yes, yes. And if it isn't there, that means that there's something wrong and your cell will not progress through the cell cycle. So the S phase happens in high concentration right after the S phase of interphase. And then your M cyclin happens right after the G2 phase to promote progression through mitosis. Here's another visual of our cyclins. Each one of these cyclins is shown as like a little sickle right there. The G2M phase cyclin will turn on its CDK target, so it's cyclin-dependent kinases, and that will allow the cell to progress to the next checkpoint. As soon as that cyclin has moved through its checkpoint, it'll be broken down. So they increase in concentration and then they're broken down. Here you have one that's promoting through the G1 phase. Here you have one that's promoting through the S phase. So the concentration increases to turn on another enzyme and then decreases after that enzyme has been activated. Cyclins turn on CDKs. Let's talk about CDKs. CDKs are cyclin-dependent kinases. They also promote going through the cell cycle. So they expedite mitosis. They want mitosis to continue. If they are not active, that means that mitosis pauses. Kinases as a whole group are going to be enzymes that phosphorylate a target. By phosphorylate, we mean add a phosphate to. So these CDKs are inactive until there is enough cyclin. And when there is cyclin, the CDK becomes active. And what it does is it adds phosphates to target proteins. And when those phosphates are added to the target proteins, the cell is allowed to go through the next step of the cell cycle. So it is promoting progression. We also have tumor suppressor genes. A tumor suppressor gene is a piece of DNA that codes for a protein that controls cell growth. And because it controls cell growth, it suppresses tumors. So a gene controls for proteins. Tumor suppressor genes suppress tumors when they are working correctly. A tumor suppressor gene halts progression, and that's because you don't want uncontrolled cell growth. Here's the way to visualize it. If my tumor suppressor gene is working functionally, I'm creating proteins that are going to inhibit abnormal cell growth, and my cell division process is under control, it's being regulated. If the gene gets mutated, and these mutations are associated with a high risk of cancer, so if I have a mutation in this gene, that means that I might have a non-functional or a defective protein. And if my protein created by this gene is defective, I could have cells that have uncontrolled growth, and that would lead to a tumor. So my tumor suppressor gene must be functional to suppress tumors. We have a couple really cool types of tumor suppressor genes. P53 is one of the most common, and P53 halts the cell cycle. It stops mitosis from happening. What it does is it looks for damage in the DNA, and if it finds it, it makes the cell cycle stop. What's really cool is not only does it make the cell cycle stop, but it will look for DNA repair. And if it can fix it, then the cell cycle can restart. But if it can't fix it, then it will tell that cell no, and the cell will do apoptosis, which is programmed cell death. In apoptosis, the lysosomes in the cell, those digestive sacs, just open and they auto-digest the cell from the inside out, and everything that was in the cell gets broken down and the components get recycled to make new cells. So when P53 is working, any cell that has an issue is identified and paused until it's fixed or paused until it's destroyed. Now, if there is a mutated P53, a mutation in the gene for P53, the protein, then you have a cell cycle that won't stop if there's DNA damage. And that means that damaged DNA will increase and increase and increase, and that could lead to a cancerous cell or a tumor growth. We also have P21. P21 helps P53, and P21 is like a supporter. Whenever P53 levels increase, P21 levels increase too, and it helps produce inhibitors for CDKs. So cyclin-dependent kinases, they allow progression through the cell cycle. When P53 sees DNA damage, it activates P21, and P21 inhibits CDKs so that you can stop the cell cycle and hopefully fix whatever problem P53 found. Our last example of a tumor suppressor gene is RB. RB is really cool because it's looking at cell size. If you have cells that are not big enough to divide, then they're going to end up with issues, and that could become problematic and relates to malfunctions in cell function, which that's fun sentence. So cell size is important. The way that RB works as a tumor suppressor gene is the gene codes for this protein RB, and RB holds onto a transcription factor. When RB is active, it's holding onto it, and so no transcription is occurring. When the cell is big enough, an enzyme will come and phosphorylate RB, and when it's phosphorylated, it drops the transcription factor, and that means that now this gene can be transcribed and used, which means that you can go ahead and advance through the cell cycle. So the size of the cell depends whether or not the transcription factor is available, and that's controlled by a gene that creates the protein RB. Here's a visual of how beautiful and complex it actually is. I don't want you to memorize anything here, but I do want you to look for words that you just learned, like CDKs and cyclin and spindle, and the different checkpoints. And note that each one of these molecules is dependent on different molecules, and there's multiple pathways to get into or out of advancing through the cell cycle. It's a new area of science, and there's going to be a lot of really cool discoveries in this because we're still learning how this all occurs. And that brings us to the mitotic index. The mitotic index is used to figure out the rate of cell division, and we use it in the lab to determine whether or not a tissue is cancerous. So what you do is you take a sliver of the tissue and you look at it under the microscope. So you're actually visually looking at the cells, and you identify what cells are doing mitosis and what cells are in interphase. If your cell is doing mitosis, you can see the condensed chromosomes. And if it's not doing mitosis, you can see an equal spread of DNA, and the nucleolus will be visible. So here I have a single cell. I can tell it's a plant cell because there's a cell wall. And here is the nucleus. It looks pretty evenly spread of DNA. These two dots are the nucleolus. If you can see the nucleolus, then you know that your cell is in interphase. So all of these cells are in interphase here. When the cell is doing mitosis, the chromosomes will condense. So this is condensing chromosomes. This one is in prophase. This is condensing. This is prophase. If the chromosomes are lined up in the center, that's going to be metaphase. So this one's almost in metaphase. And then you can see the chromosomes being pulled apart in anaphase here and here. This is anaphase occurring. This is anaphase occurring too. And then lastly, it looks like these are pretty far apart, and you can start to see the cell plate being formed between it. So I would say this is an telophase, and the cell plate is occurring. So cytokinesis is happening there. It looks like these two just divided, but I can see a cell wall between them, so I would count those as two separate cells. This one looks like telophase to me, though, because I can still see the condensing of the chromosomes, and the cell plate is pretty faint. It might not be a full cell wall there, but this is almost done. It's almost back to interphase. So you take the number of cells in mitosis divided by the total number of cells. Let's practice it. Here, imagine that someone hands you an onion and you want to plant it in your garden. But before you do, you need to determine whether onion roots grow from the base of the root, which is up here, where it attaches to the onion, or from the tip of the root, which is down here toward the letter A. To figure that out, you take a little cross section of each and you look at it in the microscope and you calculate the mitotic index. Here's what you see in the microscope. This is the root tip, so further from the onion bulb, and this is the root base right up next to the onion bulb. So take a moment, pause the video, count the total number of cells, and count the number of cells in mitosis, and then your mitotic index is mitotic cells divided by total cells. Go ahead and calculate them for each and see which one has a higher number of mitotic index. And the answer is roughly, because it's kind of hard to determine, did you count this cell or did I? But loosely, I found 20 cells divided by 75 total for a mitotic index of 2.66. And over here, I found roughly 72 cells total and 7 cells doing mitosis. And if you found 8 or 9, whatever, it's totally fine, because maybe you counted this cell and decided it was an interphase, for a mitotic index of 0.09. So we have way more mitosis occurring in the root tip. That's going to be the area of the root where your onion is growing. Let's practice it again. So here we have a type of cell. We have the number of those cells in interphase, number of cells in prophase, metaphase, anaphase, and telophase. And to calculate the mitotic index, you're going to add up all the cells in mitosis divided by the total number of cells. So note that you're not doing 1 divided by 19, you're doing 1 divided by 20, because you have total cells 20, one of them in mitosis. You're going to do it for a normal lung, a cancerous lung, a normal stomach, a cancerous stomach, a normal ovary, and a cancerous ovary. And you're going to see how the mitotic index is different between these tissues with cancer and without. Pause the video, go ahead. And did you get roughly these numbers? So what's the trend? The cancerous tissue for all types has a higher mitotic index. There's more cell growth occurring in cancerous tissue, and that makes sense for us because the definition of cancer is uncontrolled cell growth. Which cells do you think would have the highest level of mitotic index based on how often they divide? You have how often the cell divides, constantly, every few months, or only when damaged, or once in a lifetime. And then you have the type of cell here. Which one do you think has the highest mitotic index? You are correct. It is any cells that divide constantly are going to have more mitosis occurring. And I think that feels intuitive, but it's kind of hard to think about a normal cell having a high mitotic index. So in the last example, we talked about a high mitotic index, meaning cancer. That's not true because our skin and hair have a high mitotic index. It's higher than average, which indicates cancer. So in a skin cell, I would have a high mitotic index, but in a cancer skin cell, I would have an even higher mitotic index. So it's higher than average, not just high in general. Have you heard of BPA? BPA is an additive that has been put in plastic, and BPA makes plastic more durable and more resistant to scratching. BPA can leach from the plastic into your food or your water, and it has been found in 93% of humans that have been tested for it. The CDC says that the levels of BPA found in food are safe, and several studies have shown that BPA is not harmful in low doses. However, BPA got a lot of publicity probably like 20 years ago. I'm not sure if you remember it, but it was being blamed for giving certain celebrities cancer, and it was the whole media circus. And there were follow-up studies in which people gave high concentrations of BPA to animals and found that they developed cancers and tumors like gratuitously. And so the concern about BPA being cancerous is there. And because of that, majority of our plastics no longer have BPA in them. So here you can see this Nalgene bottle says BPA free. And that's because of the public being upset about it. There are still additives added to the plastic to make it durable. They're just not using BPA anymore. And so I have a study for you that I want you to check out that's really interesting. Rats were exposed to high levels of BPA, and then they were tested to see how many tumors they developed. Now, this graph is set up weird. So take a moment, look at it, read everything, and then I'm going to ask you a couple questions. Okay, if you need more time, pause it on it. Based on the data presented, which rats would you expect to have a higher mitotic index? Our graph here goes percent of tumor-free rats, 100%, everyone starts without tumors. And then over time, in days, the number of tumors that are forming causes a decrease in the graph. So if this is tumor-free 100%, 0 will be tumor-full, right? So whoever's line is more starkly decreasing is having more tumors, and more tumors would be more mitotic index. And so we would expect a higher mitotic index in the BPA group of rats. What was the independent and dependent variable in this experiment? The independent variable is what you changed. That was BPA exposure. And the dependent variable is what you're measuring, which is tumor rate. Now, I want you to critique this graph. It's not great, and it's really important to think about how we could be making experiments better. So come up with some weaknesses and some strengths. and identify what information is missing. Go ahead and pause the video for that. For me, I would change it so that it's going from 0 tumors to 100% tumors, not 100% tumor-free to 0% tumor-free. I would also edit it so that it's more clear when the tumor on average is showing up. So we usually make graphs that have averages of data instead of raw data because it makes more clear to see the trends. I would also add a figure caption so that it's really clear what's going on, because this is hard to read and hard to figure out. By convention, we usually put the independent variable on the x-axis and the dependent variable on the y-axis. But here we have two dependent variables being shown. We have the percent of tumor-free rats and the days. Both of those are things that we are collecting data on. The independent variable, the things that we changed, was whether or not the rats were exposed to BPA. And so I would edit it to have it one independent variable and one dependent variable, and maybe make two different graphs to show the two separate ideas separately. Hope you had a great rest of your day. Max says to email me if you have any questions, and I will talk to you soon. Bye.